right, in the next exam, there are really two topics that we're going to cover more than anything else, and those are membranes and gene regulation in eukaryotes, specifically eukaryotes. I think I will put gene regulation in bacteria in the next exam. So we kind of covered, you have seen one video where we talk about histones and things like that, and CpG islands and turning genes on and off and stuff like that. That will be one major subject. The other subject is here, and we've kind of covered it a little. We've talked about membranes and kind of their structure a little bit, but we're going to go into a lot more detail now about how membranes work and what they do for you. So for the first thing, right here, find this table in the book and make sure you know each of these. I may or may not ask you to list these, so I would memorize them, I guess, each one of these, and then I'm going to go through these and give you my kind of lecture on these, and then I'll go back and give you a PowerPoint lecture. But I want to talk about each of these separately, so that you can get a kind of a overview of what each of these is and how the membrane is related to each of these. So let's start with the first one here, the selective uptake and export of ions and other molecules. Other molecules can be things like proteins and carbohydrates. It could be lipids. It could be anything. Ions, of course, are just charged particles like protons or electrons. But molecules, this can pretty much be anything, and they can be really large. These are tiny. These are small. So selective uptake and export. So and all that's saying is getting in and out of the cell. So they're just using big words to kind of say something very simple. One of the functions of biological membranes is to get the stuff you want in and out of the cell. It can be tiny things like ions or larger things like molecules. And that's all this sentence says. But let's draw it. Let's kind of get an idea of what they're talking about. In other words, I'm a cell. Let's say I'm a bacteria. It could be really any cell. It could be one of your cells or a bacteria. We just need to put a cell with a membrane around it. And the whole idea is there are things that we want to get in, all right? And there are also things that we want to get out of the cell. Easy enough. Well, there are also things that are in the cell that we never want to leave. All right? We want them to go up here and never get out. All right? There are also things on the outside that we want to never get in. Say a virus or something like that. Doesn't always work that way. But the whole idea is selectively means we get to choose. The cell wants to say, okay, these things are in here. They took a lot of energy to build them. We don't want them to ever get out. All right, something like that. Or it's food. We don't want that escaping. Then there are things that might be dangerous. We don't want those to get out. Now, what kind of things do you want to get out? Well, maybe you're sending a signal to another cell if you're a eukaryote. But you could also be sending signals to other bacteria if you're a bacteria. So there are some things that you want to get out. Maybe it's a waste. You want to get that out of the cell. All right, so there are things you want to get out of the cell. And then there are things you want to get in. Maybe you want to get in those signaling molecules from the other cell. So you are sending a signal to one cell. Well, the other cell has to be able to receive it. You also want to get things like food inside the cell. So wastes you want out. Things that you can use, glucose for energy. Let's see, what else would you want in? Well, you'd want any type of amino acids and things like that you could bring in, carbohydrates, which would be glucose. You might want to bring them in and digest them into glucose. Then let's see, what else? Fats, lipids, you could bring those in from outside. So the whole idea, water, sometimes you want to get rid of water and sometimes you want to keep it inside. So this works both ways. Sometimes you want to get rid of the water, sometimes you want to bring the water in, sometimes you just want to keep keep it in. That would be the same as trying to keep something in. Sometimes you don't want the water in. So the whole idea here is the cell, this membrane is selectively permeable. So let's go back now to the list. So the selective uptake and export of ions and molecules. So ions are like electrons, protons, that sort of thing. Molecules can be anything. It could be a proton, a signaling molecule that you're sending to another cell. A hormone. One cell wants to give a chemical signal to another cell. That's a hormone. All right, so you might want to release that. It's a very complicated and expensive molecule to make, probably. And you want to make it, you want to get it out, and you want to get it to the other cell. That's a large molecule. And there are going to be, what we're going to find is there are different ways to get things in and out of the cell. Some things use diffusion, all right, of ions. So we'll learn about diffusion in another video. But there's osmosis, that's good for water and things that can dissolve in water. Then there are large things, these larger molecules, which we're going to have to create pumps and pores to actually pump something out of a cell or bring it in. And of course, that's going to involve proteins, because all the work done by proteins, there are going to be proteins that are in the membrane. There are going to be pumps and pores for letting specific things in and out, very large things. All right, over here, these are small things. They can diffuse, which means they can pass in between the lipids in the membrane. But then there's like water and things like that, which can move via osmosis, depending on what's dissolved in the water. And so we'll have to talk about all of those things. But you need to understand the selective uptake and export of ions and molecules. Now, cell compartmentalization, we've talked about that early in the course, but let's go look at that again. All right, so let's see, let's get rid of most of this. That red line does not want to disappear. Let's see if I can use an eraser. There we go. All right. So, there we go. It's got a hole in the cell. All right. So let's go back, and we will take our cell, and what we're going to do is, like I said, we are looking for cell compartmentalization. So this is something that a bacteria, if this is a bacteria, it doesn't have any compartmentalization. This is like the idea of rooms in a mansion versus an efficiency apartment or a motel room. A little bacteria doesn't have any compartmentalization, just like a little motel room doesn't have a lot of rooms. So everything has to be done in the same place. So very little compartmentalization in a bacteria. But if this is a eukaryotic cell, what do we have? Well, we have organelles, all right, like the nucleus, and which will have what, chromosomes in here. All right, I'll draw a couple of chromosomes. All right, so we've got 2 pair of chromosomes. So this is now a eukaryote. It's not a prokaryote. And there is a membrane around the chromosomes here. All right, and this is the nucleus, of course. And it's very similar membrane to the membrane on the outside. The differences will be what's kind of floating in the membrane. So I'm going to draw some of these. What you'll get are little proteins that are sticking into the membrane. They're maybe sticking out on both sides like I have it here. Maybe they just kind of float on top. Maybe they only float and they point inward. So there are all these little proteins that we will find, all right, scattered along the outside of the cell, all right, here. But there are proteins in the nuclear membrane as well. And that's going to control specifically what gets in and out. Depending on what proteins are here, a lot of these are what we call pores and pumps, which I will go into more detail later. But these pores and pumps specifically allow certain things in and also might pump certain things out. And that gives you the unique chemical characteristics of your organelle. So inside the nucleus, you are trying to keep the chromosomes safe. So you don't want anything very harsh in here. You're not going to want it to be very acidic or basic. You're going to get very neutral, very water-like. All right. You're going to try to keep it as literally as fewer reactions as possible, I guess, or the reactions that would occur in here would be ones that would help along transcription. So the whole idea here is mostly we're doing transcription in here, and if that's the case, you want the chemical environment to promote transcription. Now there might be other organelles, all right, and maybe this is a peroxisome. I'll draw a peroxisome like this, just to make it look different from anything else. So kind of stripes. So a peroxisome, its function is primarily to digest things, break them down into simpler things, and often did I say peroxisome or lysosome? That's a lysosome. Would be to break things down. Peroxisome is to detoxify. But either one, let's do the other one. All right, so the other one, I'll make it more solid. So here is a slightly different organelle, and it's a compartment. And the chemistry inside these are going to be different. So if it's a peroxisome, let's say this one is the peroxisome. Peroxisome is usually there to detoxify poisons or something that might be dangerous. You want to definitely kind of put it in here, and you're going to have to deal with it specifically. Maybe it's a waste that you created doing some other reaction, and you have to get it in here immediately so that it doesn't damage anything else. The lysosome is usually for breaking something down into useful components, and so therefore you bring some type of food in, all right, something that's useful, and once you get it into the cell, you then put it into a lysosome to digest it, and then the parts that you make, maybe you break it down into two different things and you want those in the cytoplasm, you break them down and then pump those two different things out. Or maybe you keep one in. Maybe one of them is poisonous and you want to ship it over here to a peroxisome. That could happen as well. There are methods for that to happen, for this to send something over here. So there would have to be, to have all this to occur, there would have to be a pump or a pore to let whatever this is into the cell. Then there would have to be a pump or a pore that would let that actually be transferred into this organelle. Then you would have enzymes that would break it down. Those are proteins. So there had to be a way, a pump or a pore perhaps, to get the enzymes in here. So there are different types of pumps and pores that are going to only let certain things in. So this one let whatever it is through, and then here it let it, maybe the same one that was made, that created here a set of proteins that act as a pump or pore are placed into the membrane that let whatever this food is in, let it in, and then it gets pumped through, and then maybe it reaches this organelle, and the same set of proteins are in this membrane, and it gets pumped in. And then there would be some enzymes, some proteins that need to get pumped in that can digest it. And then you would need, again, you're going to need some pumps or pores. Whatever the products of that are, you're going to have to pump those out to get them wherever they need to go. Maybe you broke them down into everything down into ATP, and you want to pump that back out into the cytoplasm. And then maybe you had some leftover parts, some amino acids or something that you wanted to pump out so something else could use them. Maybe to make proteins, a ribosome could perhaps utilize that. So you pump some ATP out, and then you pumped out some amino acids to form a protein at a ribosome. But you'd have to have different pores or pumps to get these things across the membrane in each case. So all along here, you would have to have these things built and designed and ready to go to do this and have this little compartment function. Same over here with this one. You would have to have things that could let things in and, then push things out, whatever needs to be pumped out. Usually you pump things out and you let things in, but you never know. It could be pumps on either side. But this is the kind of the idea here behind membrane transport. And so we're going to learn about those different types of transports, pumps and pores, osmosis, diffusion. All of those are ways to get things across a membrane and either into the cell or into an organelle or out of the cell or out of an organelle. All right, so keep all of that in mind as we move forward. All right, so we talked about compartmentalization. All right, and compartmentalization is just this idea that organelles. Organelles are the compartments that you create in cell compartmentalization. Realize that bacteria don't really do this. Maybe they can create a little organelle to store some water or something. We'd call that maybe a vacuole, but it's not a complex structure. The complex structures that we use, all right, are much, they have all these pumps and pores and things. When I say we, I'm talking about eukaryotes. They have lots of complex protein complexes where a bunch of proteins come together to form these pumps and pores. So just understand that that is cell compartmentalization. Protein sorting we will get to in the PowerPoints. I'm not going to make a drawing for protein sorting. All right, anchoring of the cytoskeleton, I will also wait until we get to the PowerPoints to do that one. Now, the production of energy intermediates such as ATP and NADH. I'll talk about it briefly, but not too long here. This is simply the idea. I'll kind of remind you this one. I can quickly do this one. That when you bring things in like carbs, carbohydrates, all right, into the cell, you usually are going to have to break them down into, at some point, glucose. Just to remind you how this works. All right, and to do that, okay, maybe we did it in here. We pumped these carbs inside, we allowed them in here, and then we pumped them into this, say, lysosome. And in that lysosome, there were some proteins that were made and pumped in there as well. And those allow you to break the carbs down into glucose. So there would be an enzyme of some sort. It's a protein. We just call an enzyme here in this case. All right, that would be needed here, like a chemical reaction. All right, you need carbs plus the enzyme to produce the glucose. And then, of course, you want to get the glucose out once it's made. So this is it. Now, at one point, what do you do after you have glucose? You almost always, you could store it, but you almost always turn it into ATP, adenosine triphosphate. Now that can occur sometimes within the cell or the cytoplasm, but a lot of times it occurs in mitochondria or chloroplasts. And there will, of course, be many proteins that are needed to do this. All right, so you'll have a set of enzymes, proteins, that carry out several chemical reactions in an order. And we usually call this a biochemical pathway. And I might give you another video on something like that. But there would be several of these proteins that in a series of steps you might convert something like glucose into ATP. All right, now once you have ATP, if you don't remember, is what powers your proteins. So your proteins have a shape, all right, and that shape gives them several, both structure and chemical capabilities. But to do work, they usually have to change shape. So it has this shape, and now it has this shape, very different shape. And to do that, you usually, how we draw it is you draw it as you are converting ATP into ADP. And this releases energy. So when this occurs, you add energy, and I'll draw that as a big E, meaning energy, to the protein. And so that energy, when you convert ATP to ADP, converts this protein from this shape to this one. It made it do physical work. Now, what is ATP versus ADP? Well, this is adenosine tri, meaning 3 phosphate. Adenosine triphosphate means it has 3 phosphates stuck onto an adenosine. So without going into a lot of chemical structure, just think of it this way. It's an adenosine with 3 of these phosphates. I'll draw those as P's on here. And what you do is this chemical reaction that involves this protein kind of has the ability to rip one of these phosphates off. So it tears it off. And when it tears it, it breaks this bond right here. And that bond, when it breaks, is the energy that is released to the protein. So in the end, what do you get? You actually get ADP, plus you get actually something called inorganic phosphate, PI. That's kind of how you draw it. It's a P and a little I. And the PI is usually considered a waste, and it might be pumped out of the cell. It actually gets recycled quite a bit. The ADP is actually recycled also at the mitochondria or chloroplast. So whether or not you really consider them wastes, like things to get out of the cell, maybe not, because if you had a bunch of it and you needed to get rid of it, yeah. But a lot of times it's recycled. And there are cases where we will see that the P is actually stuck back onto the ADP to make new ATP. But we'll get there at a later date. We won't talk about that yet, how that works. But we do need to know that you rip the P's off to release the energy, and that energy goes to the protein, which allows it to change shape and do its job, whatever it is. And there are proteins all along here, maybe 3, 4, 5, maybe a dozen, all right, that might go to convert something like glucose to ATP. And then, of course, you want to pump that ATP back out in the cell so all your proteins will work. And so understand how this works, and understand that there are pumps and pores, and don't forget how ATP is the thing that powers your proteins, all right? It powers your cell. And I think I've said it before, you can think of ATP. ATP is to your cell what gasoline is to a car. Without ATP, nothing runs, all right? Those proteins have to change shape to have the cell run. All right, so now let's go and see what our next topic is, which I've already talked about, cell signaling. Cells can talk to one another, all right? So we have a cell, and we want to talk to another cell over here. So let's change the color here. So there's another cell somewhere over here, and we want to talk to it. Well, that means, of course, we need to create a protein or something that we can pump out, all right? And then this one, of course, needs to be able to pump it back in, and that is a chemical signal from this cell to the other. So cell signaling, very simple, not a big deal, but you will need these pumps and pores. You'll need one here to pump it out, and you'll need one here to let it in. And we'll talk more about the details on that as I go through these lectures for this next exam. All right, so cell and nuclear division. All right, I'm not going to talk a lot about it, but the whole idea is membranes and proteins are needed to basically duplicate the chromosomes and to divide, all right, the nucleus so that you can actually split the cell into two daughter cells. And there are, of course, membrane involved in all of this, because you're going to have to create new membrane, all right, to duplicate the daughter cell, all right, and create a nucleus there and all the ribosomes. And you're going to have to do all of this. Proteins will do all the work, but you're also going to have to produce some lipids and some extra membrane to get all of this done. And that's all the important function is to, it is a part of cell and nuclear division, because you're going to have to move the membrane around and you're going to have to make new membrane and stuff like that. So it is a part of this table. Now, adhesion of cells to each other and the extracellular matrix. So I will explain here the extracellular matrix in a second, but realize a lot of cells stick to one another. So let's talk about that. So let's just get a new picture here. All right, let's zoom in. There we go. So the first thing, cell adhesion. So some cells, let's say these are skin cells. Well, skin cells are going to be packed up next to each other. And they don't just stand there normally, I mean by themselves. Or maybe this is the lining of your stomach. Those cells are packed very tightly together. And so there has to be proteins and things that go from one cell to the other to kind of like a staple, I guess you could think of it that way, that are going to kind of connect these two, all right? And it isn't just two, you know. And they're going to keep these from basically wandering off to wherever they're, you know, or floating off, wherever they're on. You want them to be connected. And so there will be proteins in each cell that are in the membrane that are going to make them clingy, all right, and stick to each other. Now, there are also some carbohydrates usually involved. Carbohydrates tend to be sticky, and so you'll have a protein with maybe a carbohydrate stuck on the outside. So you might have the outside of the cell here, and there's a protein or a bunch of proteins stuck in the membrane. And then on top of that, you'll have a little carbohydrate. And that little carbohydrate will be kind of sticky like Velcro. And then the other cell is up here, and it'll do something very similar. All right, it'll have a protein or a bunch of proteins, and it'll also have some little carbohydrates that are stuck out here that act like Velcro, and they come in there and they touch the other side, and essentially they stick. And so these can be held together. So that kind of gives you the idea behind adhesion of cells to each other. But there's also this thing called the extracellular matrix, which most people don't know about. So we need to learn that. It'll definitely be a test question. What is the extracellular matrix? And let's get a new one again, and we'll discard that one and view. Zoom in. All right, so let's make ourselves a cell, some sort of cell here, and a cell here, and a cell here. And believe it or not, skin and things like that do have cells that are right next to each other. But a lot of your tissue, like your brain, for instance, or some other organs, they are not cells just crammed up next to each other. They're actually spaced out. And when you touch them, I mean, skin is the exception. Skin really is, the cells are kind of packed next to each other. But internally, your cells really aren't packed next to each other. They are separated by quite a bit of distance. And the stuff in between them is something called the extracellular matrix. And it's like a goo. All right, it's more like Jello. And in it there are fibers, all right, and tunnels. So there might be some fibers in here, or some things that would produce tunnels. Let's see, roughly. But think of it as being jello with holes in it. All right. And basically, the cell produces, all the cells here produce this material. So the cells are releasing, as we've seen, they can pump things out. They're pumping chemicals out, all right, that actually produce this goo, kind of a semi-firm stuff that produces what is called the extracellular matrix. Extra meaning outside or beyond or around, and matrix. And so this is a matrix in the sense of it's got some solid pieces in it, and they're usually in the form of some fibers that give it some toughness. All right, so it's not, and then there are proteins and there are tunnels in it, and it's got a lot of stuff in it. There are people who just study just the extracellular matrix, but there's a lot of water in here, and it's, like I said, similar to jello maybe. And really, these cells in most of your body, most of your organs, the cells are quite far apart, and you're just used to things like the skin. But a lot of the time, an organ, like a, they take out your spleen and the stuff that makes up the spleen. It's really, the cells are kind of far apart, and they're sitting inside this matrix, all right, the extracellular matrix. And the chemicals they send are, have to travel out of the cell and then make it to the others, and they have to travel through this matrix. And things just kind of float around in here. And interestingly, now your cells are large. A little bacteria might be in here, and it can maneuver quite well. The bacteria are much smaller than our cells, and these bacteria will get in here, and they'll just kind of scoot around and move around things, and they are just a general nuisance. Your body, believe it or not, your body doesn't pay any attention to them unless they start altering the chemistry out here. So you might have a lot of bacteria everywhere in your body, but as long as they're usually not causing a disturbance, your body doesn't react too strongly. But they start messing with things or growing too large. Most of them can't live inside your matrix very easily, and they may get in there. They may not die right away, but they don't reproduce very well. But if you get some bacteria in there that can actually thrive inside your body, inside this matrix, and they start growing, that's when your body starts to notice them. Mainly, they'll start producing a lot of wastes, and that will signal to your body that you need to go take care of this. And these bacteria, if they don't grow and don't multiply too much, your body doesn't really pay much attention to them. But if they're the type that can grow inside your matrix, then your body will actually do it. So you need to know about this extracellular matrix. You need to know that a lot of your organs are actually not just the cells, but they're this stuff. Actually, when you touch it, you touch an organ, and the spleen or something, and or even the stomach. You're not really touching the cells usually. You're touching this kind of goo with all these fibrous kind of things in there, and there are globules of all kinds of things. There's just a ton of things in the matrix that we don't have time to go over. But you're not really touching the cells. The cells are within the matrix, and the matrix creates kind of a firm support. Like I said, it's not real tough, but it's tougher than just the cells. The cells are actually pretty wimpy. They're just membranes filled with cytoplasm and other organelles. Or organelles within the cytoplasm, so more membrane and more cytoplasm. There's not much to a cell. And to support and give physical support and structure to an organ, you actually really need to make the matrix around it. The exception is kind of like a skin cell. Skin cells are on the outside, and they're actually usually dead, though. You have like several layers of dead skin cells on the outside of you, and they're actually very useful. It keeps bacteria out. But the idea here is this is our our last thing here, adhesion and the extracellular matrix. So you need to know about this extracellular matrix. You need to know the concept of adhesion. You need to know all of these things. I've covered the main ones that are not covered well in the book, I don't think. And I'll cover the other things here as we get through these PowerPoints in the next lecture.